The overall goal of this procedure is to detect self-reactive T cells with a high degree of specificity using MHC Class II Dextramers. In this video, cerebrum is harvested from the brain of a myelin proteolipid protein-induced experimental autoimmune encephalomyelitis mouse. This is accomplished by first carefully harvesting the tissues and preparing the agarose blocks. The second step is to cut the agarose-embedded tissue into 200 micrometer thick sections with a vibratome. The sections are then transferred to tissue chambers in a 24-well plate, where they are stained and fixed. The final step is to mount the tissue sections on microscopic slides. Ultimately, confocal microscopy is used to show the cells bound with Dextramers and CD4 antibody.
We first conceived this idea when we first demonstrated that the MHC class II Dextramers were more sensitive than the conventional MHC class II tetramers for detecting the antigen-specific self-reactive CD4 T cells.
Generally, individuals new to this method will struggle because sectioning of fresh tissues and their handling is fairly challenging.
Visual demonstration of this method is critical, as the sectioning, mounting, and analysis steps are difficult to learn because they are tricky. In addition to Ting, two postdoctoral research associates, Chandi and Arun from my group, and Christian from Dr. Zhou's laboratory will be demonstrating this procedure.
Following the euthanasia of experimental